The seafloor here is silent in a way that feels absolute. More than 10 kilometers below the surface, light has never reached this place. The water presses inward with a force that would crush steel, and the temperature hovers just above freezing. Nothing moves quickly, nothing wastes energy, and yet, spread across the sediment like abandoned architecture, something vast and alive quietly reshapes the rules of biology. At first glance, it looks like debris-tangled threads, brittle fans, branching shapes scattered across the mud. Some are the size of dinner plates, others stretch wider than a human body. For decades, remotely operated vehicles drifted past them, cameras recording what scientists assumed were sponges, corals, or mineral formations. They were logged, labeled, and largely ignored. Then samples came back to the surface. What researchers found inside those fragile forms was not a colony of animals, nor a plant, nor anything that fit comfortably into familiar categories. It was a single cell, one cell, organizing itself into complex, sprawling structures that rival the size and spatial presence of multicellular life. A single cell capable of building a body without tissues, organs, or symmetry. A cell that thrives where almost nothing else can dot down here. Survival is not about speed or strength. It is about patience, chemistry, and an intimate relationship with the seafloor itself. Food arrives slowly, drifting down from the sunlit world above as marine snow-dead plankton, fecal pellets, fragments of life. For most organisms, it is barely enough. For this organism, it is everything. These giant single cells collect sediment, trap particles, and incorporate heavy metals that would poison most life. They reshape the chemistry of the mud beneath them. They host entire communities of microbes within their structure. They leave behind tracks and imprints, not through movement, but through presence, through the simple act of existing. In one place for decades, nothing about them aligns with what biology textbooks once taught about the limits of cellular life. Size, structure, longevity. Even the boundary between organism and environment begins to blur. Today, we're diving into Xenophyophore, some of the strangest life forms ever discovered, and why their existence forces scientists to rethink what a single cell can be, how life adapts to extreme isolation, and how much of Earth's biology remains hidden in the dark. Because in the deepest parts of the ocean, where pressure erases mistakes and energy is scarce, evolution has written a story that doesn't follow the rules we thought were universal. The descent into the deep sea is not a journey through space, but through constraint. With every meter downward, options disappear. Light fades, temperature drops, and pressure mounts until survival becomes an exercise in biochemical precision. By the time the seafloor comes into view, life is no longer shaped by competition or speed, but by endurance. This is the Haddle and abyssal realm, vast plains and trenches that make up most of the planet's living space, yet remain largely unexplored. In places like the Mariana Trench, the ocean floor lies deeper than Mount Everest is tall. The water above weighs more than 1,000 times the pressure at sea level. Any air-filled space collapses instantly. Proteins misfold, membranes stiffen. For most organisms, the physics alone is fatal. Food here does not grow, it falls. The deep sea survives on what the surface discards. Microscopic plankton die, sink, and fragment as they descend. Fecal pellets rain down from grazing animals above. Occasionally, a whale carcass plunges to the bottom, creating a brief explosion of abundance before vanishing into scavengers and microbes. Between these rare events, the seafloor exists in a state of near starvation. This scarcity rewrites evolutionary priorities. There is no advantage in being fast if there is nowhere to run. There is little benefit in aggression when encounters are rare. Instead, success belongs to organisms that can wait, that can extract value from sediment grains, that can endure years, sometimes decades between. Meaningful meals. The seafloor itself becomes part of the biology. It is a chemical archive layered with iron, manganese, uranium, and other metals that leach slowly from seawater. Oxygen barely penetrates its surface. Microbial communities dominate, breaking down organic matter molecule by molecule. Any organism that lives here must negotiate directly with this chemistry, not just tolerate it, but often exploit it. Most deep-sea animals meet this challenge by simplifying zoometabolisms, reduced muscles, minimal movement. But some life forms take a different path. Instead of shrinking, they spread. Instead of isolating themselves from the sediment, they incorporate it. Instead of forming tissues and organs, they remain cellular yet vast. These environments do not reward complexity in the way shallow ecosystems do. They reward integration with place, stability, chemical adaptability, a willingness to become part of the seafloor rather than merely inhabit it. It is within this context that xenophyophores emerge not as anomalies, but as logical outcomes. When energy is limited and time is abundant, evolution does not rush toward familiar solutions. It explores extremes. It stretches definitions. It allows a single cell to become architecture. To understand xenophyophores, it is not enough to look at them alone. They are inseparable from the world that shaped them, a world where survival is measured not in seconds or generations, but in the ability to persist quietly, chemically, and indefinitely in the dark dot. At the cellular level, xenophyophores should not exist as they do. Biology long held that single cells face hard physical limits. As size increases, diffusion becomes inefficient. Nutrients struggle to reach distant regions. Waste accumulates. Structure collapses without internal support. These constraints explain why multicellular life evolved, why cells specialized, divided labor, and formed tissues. Xenophyophores ignore this logic entirely. They are single-celled protists, distant relatives of foraminifera, yet some grow larger than a human hand, not by becoming thicker or denser, but by becoming spatial. Their bodies are not compact masses of living material. Instead, they form elaborate branching networks filled largely with sediment particles gathered from the seafloor. These particles are glued together by organic material secreted by the cell, forming a fragile skeleton called a test. What appears to be a body is mostly borrowed matter. Within this structure, the living cytoplasm threads itself through countless tiny channels, creating a distributed system rather than a centralized one. There is no single core, no organ-like regions. The cell spreads its metabolism across space, reducing the distance nutrients and oxygen must travel at any one point. Growth does not mean thickening, it means extending outward. Following the contours of the sediment, this architecture solves problems that size normally creates. Diffusion remains effective because active regions are thin and widespread. Damage to one part does not threaten the whole. If a section collapses or is buried, the rest persists. The organism does not need symmetry, mobility, or speed. It only needs contact with particles, with chemistry, with time. Even the materials xenophyophores use challenge expectations. Their tests often incorporate heavy minerals, barite, manganese nodules, metal-rich grains that most organisms avoid. These materials add stability in a high-pressure environment. Anchoring the structure against currents and sediment shifts, what would be toxic or useless elsewhere becomes scaffolding here. The result is something that defies visual intuition, a single cell that resembles a sponge, a leafless shrub, or a shattered shell. Something that occupies space like an organism but behaves like a process. It does not hunt, it does not flee. It filters, traps, absorbs. Scientists once mistook xenophyophores for multicellular animals precisely because of this scale and form. Only under microscopes did the truth emerge. One nucleosaur, sometimes many nuclei sharing a continuous cytoplasm, a body built without cells working in teams, complexity without division, size without tissues. This forces a reconsideration of what biological organization can look like. Multicellularity is not the only path to large size. It is one solution among many. In the deep sea, where energy is sparse and stability matters more than efficiency, xenophyophores reveal an alternative strategy, one where life expands not by adding parts, but by rearranging space. They are not primitive remnants of early life. They are specialists, refined by conditions so extreme that conventional biological rules loosen their grip. For most life, heavy metals are a threat that must be avoided or neutralized. Elements like lead, mercury, uranium, and cadmium interfere with enzymes, disrupt membranes, and damage genetic material. Even in trace amounts, they can be lethal. In shallow ecosystems, organisms evolve elaborate detoxification systems or behaviors that keep them away from contaminated environments. In the deep sea, Avoidance is not an option here. Metal-rich sediments are the norm. Iron and manganese seep. From the crust, radioactive elements settle slowly from seawater and bind to fine particles. Over time, the seafloor becomes a chemical mosaic where toxicity is not an anomaly, but a background condition. Xenophyophores do not merely tolerate this environment, they incorporate it. Their bodies are filled with grains drawn directly from surrounding sediment, including particles enriched with heavy metals. These grains are not metabolized or broken down. They are stored, immobilized, and arranged within the test, effectively locking potentially harmful elements away from the living cytoplasm. What would poison other organisms becomes structural material. This strategy does more than neutralize danger. It reshapes the local chemistry by concentrating metals within their structure. Xenophyophores alter sediment composition around them. This creates microenvironments that influence microbial activity, oxygen availability, and nutrient cycling. The organism becomes a chemical filter, selectively accumulating what the environment provides in excess. Inside the living portions of the cell, additional safeguards operate. Xenophyophores compartmentalize waste products into specialized inclusions, isolating reactive substances from critical cellular machinery. Their cytoplasm contains binding proteins and molecular chelators that further reduce toxicity. These mechanisms are slow, passive, and energy-efficient, well-suited to an ecosystem where energy is the most limited resource of all. What makes this adaptation remarkable is its scale. A single cell managing chemical threats across a structure spanning centimeters, sometimes more. There is no centralized detox organ, no specialized cell types. Instead, detoxification is distributed, spatial, and deeply integrated with the organisms. Architecture, this relationship with toxicity extends beyond self-protection. By stabilizing metal-rich particles, xenophyophores reduce their bioavailability to other organisms. In doing so, they act as sinks in the deep-sea ecosystem, subtly regulating the movement of harmful elements through the food web. Their presence changes how metals circulate, not through active control, but through persistence. In environments where life is sparse, such influences matter. A xenophyophore is not just surviving chemistry, it is shaping it. This reframes toxicity itself. In extreme environments, danger is not always something to escape. Sometimes it is something to manage, absorb, and repurpose. Xenophyophores reveal a version of evolution that does not seek purity or safety, but balance, finding stability within conditions that would otherwise seem incompatible with life in the deep sea. Survival is not about eliminating threats, it is about becoming inseparable from them. Inside a xenophyophore, life does not exist alone. What appears from the outside as a fragile, sediment-filled structure is, on a microscopic level, a layered habitat. Within the branching channels of a single cell, entire microbial communities find shelter. Bacteria and archaea live embedded in the cytoplasm or nestled among sediment grains, forming internal ecosystems that blur the line between host and environment. This is not a passive accident. Xenophyophores draw in fine particles rich in organic matter, dead plankton fragments, microbial remains, chemical residues drifting down from above. These particles become food not just for the xenophyophore itself, but for the microbes that accompany them. In return, those microbes perform chemical transformations the host cannot easily manage alone. Some break down complex organic compounds into simpler molecules that the xenophyophore can absorb. Others participate in nitrogen and sulfur cycling, releasing usable nutrients in an ecosystem where fresh inputs are rare. In oxygen-poor sediments, anaerobic microbes thrive, performing metabolic processes that would be impossible in surface. Waters. The xenophyophore does not digest these communities, it accommodates them. There is no immune response to eliminate internal microbes, no attempt to sterilize the internal environment. Instead, the organism's slow metabolism and stable internal chemistry create conditions where microbial populations can persist for years. The result is a living consortium, one cell hosting many lives, each shaping the internal environment of the other. This relationship is not unique in nature, but its scale is unprecedented. Corals rely on symbiotic algae. Termites depend on gut microbes. But these partnerships occur within multicellular bodies designed for compartmentalization. Xenophyophores achieve something similar without tissues, organs, or cellular boundaries. Everything exists within a shared cytoplasm, separated only by gradients and distance. The benefits flow both ways. Microbial activity alters local oxygen levels and chemical composition, creating microzones within the structure. These zones may protect sensitive regions of the Xenophyophore cytoplasm from oxidative stress or toxic byproducts. Meanwhile, the host's structure shields microbes from physical disturbance and sudden chemical shifts in the surrounding sediment. Over time, this internal ecosystem becomes stable enough to influence the surrounding seafloor. When xenophyophores die or collapse, their structures release accumulated microbes and nutrients back into the sediment, seeding new communities. Their presence increases biodiversity in an environment otherwise defined by scarcity. This challenges a basic assumption about individuality. Xenophyophores are not solitary entities in the traditional sense. They are frameworks, single cells, that function as habitats. Their biology is not defined solely by their own metabolism, but by the processes unfolding within them. In the deep sea, where isolation dominates and encounters are rare, xenophyophores create connection. They gather life, concentrate it, and hold it in place. What survives here does so not by standing apart, but by allowing complexity to arise inside simplicity. A single cell becomes an ecosystem, not through control, but through coexistence. Time behaves differently on the deep seafloor. Without sunlight, seasons lose meaning. Without storms or tides, change arrives slowly, often invisibly. For organisms living here, survival is measured not in moments of action, but in the ability to remain unchanged for long stretches of time. Xenophyophores are built for this kind of existence. They do not grow quickly. Their expansion depends on the slow accumulation of sediment particles and the gradual extension of cytoplasm into new spaces. Each addition reflects months or years of environmental stability. There is no rush because there is no advantage. In haste, energy arrives sporadically, and every metabolic decision carries a cost. This slow pace suggests extraordinary longevity. While exact lifespans are difficult to measure in organisms that grow millimeter by millimeter, evidence points to xenophyophores persisting for decades, possibly longer. Their structures show layers of accumulated material, chemical signatures that record extended periods of growth and dormancy. Some appear unchanged across multiple deep-sea surveys conducted, years apart. Stillness becomes a survival strategy by anchoring themselves to the sediment. Xenophyophores avoid the energetic cost of movement. They do not search for food. They wait for it. Their sprawling forms increase surface area, maximizing the chance that passing particles will be trapped. Over time, this passive collection outperforms active foraging in an environment where encounters are rare. This approach carries risks: burial by shifting sediment, disturbance by passing animals, or collapse of the test can damage sections of the organism. But xenophyophores are resilient in a way that mobile life often is not. Damage remains local. The rest of the structure continues functioning. Growth resumes around the injured area, slowly incorporating new material. Longevity here is not about resistance, but repair. Even reproduction follows this temporal logic. Xenophyophores reproduce through processes that remain poorly understood, likely involving the release of reproductive cells into the water column during rare favorable conditions. These events may be infrequent, synchronized with subtle environmental cues rather than predictable cycles. Such life histories challenge assumptions shaped by surface ecosystems, where speed, turnover, and competition dominate. In the deep sea, success belongs to organisms that minimize change, that invest in persistence rather than proliferation. Xenophyophores embody this philosophy at a cellular level. Their stillness leaves marks, tracks in the sediment, subtle alterations in chemistry, enduring structures that become landmarks for other organisms. They shape their environment not through action, but through duration. In a world where most life is defined by movement and response, xenophyophores remind us that survival can also mean staying, enduring, allowing time itself to become the primary ally on the deep seafloor. Where almost nothing happens quickly, life learns to measure success in years, not moments. For much of modern biology, complexity has followed a familiar storyline. cells became cooperative. Cooperation became specialization. Specialization produced tissues, organs, nervous systems. Large size, intelligence, and ecological influence were seen as outcomes of this upward ladder from single cells to intricate multicellular life. Xenophyophores interrupt that narrative. They demonstrate that complexity does not require multiplication. It can arise through arrangement, through scale, through relationships with environment, rather than internal division. A single cell, given enough time and the right conditions, can occupy space, shape chemistry, host other life, and persist. Across decades without ever becoming multicellular, this forces a reconsideration of what evolution optimizes for. In energy-rich environments, efficiency matters. Rapid growth, specialization, and tight regulation are rewarded. In energy-poor environments like the deep sea, those same traits become liabilities. Tissues require maintenance. Organs demand coordination. Nervous systems burn energy continuously. Xenophyophores bypass these costs entirely. Instead of internal complexity, they externalize it. Their bodies are mostly sediment. Their structure is partly environment. Their function emerges not from specialized parts, but from geometry, chemistry, and time. They rely on gradients rather than circuits, on diffusion rather than transport systems. Evolution here favors designs that blur the boundary between organism and habitat. This has implications beyond the deep sea. Early life on Earth likely existed in similarly harsh, low-energy conditions. Before oxygen-rich atmospheres and abundant food, survival depended on patience and chemical adaptability. Xenophyophores may resemble strategies that were once common ways life explored size and influence, long before multicellularity became dominant. They also challenge how scientists define individuality. If a single cell can host entire microbial communities, shape ecosystems, and persist independently, where does the organism end? Is complexity measured by internal parts or by ecological role? Evolution does not move toward a single ideal form. It explores possibilities, abandoning and revisiting strategies as conditions allow. Multicellularity is one successful outcome, not an inevitable destination. Xenophyophores represent a parallel solution, one that thrives precisely because it ignores the path most life took in doing so. They remind us that the history of life is not a straight line, but a branching landscape filled with experiments. Some become dominant, others remain hidden, quietly successful in places few humans ever see. Understanding xenophyophores means letting go of assumptions about progress and evolution. It means accepting that simplicity and complexity are not opposites, but tools deployed differently depending on where life finds itself. In the deepest parts of the ocean, evolution did not climb upward, it spread outward. Even now, xenophyophores remain only partially understood. They are known mostly through fragment samples, brought up by submersibles, images captured briefly by drifting cameras, chemical signatures left in sediment cores. Entire aspects of their biology remain inferred rather than observed. How they reproduce with such infrequency, how they coordinate activity across a body without internal compartments, how they respond, if they respond at all, to slow environmental change. Their obscurity is not accidental, it is structural. The deep sea floor is Earth's largest habitat, yet it is sampled unevenly and rarely revisited. Xenophyophores live in places where research is expensive, logistically difficult, and slow. Years can pass between observations of the same site. In that time, these organisms may have grown, altered their structure, or quietly died without leaving obvious traces. What they reveal is not just something about themselves, but about the limits of human knowledge. Biology has been shaped largely by what is visible, accessible, and fast. Laboratory organisms, coastal ecosystems, species that respond quickly to manipulation. Xenophyophores resist all of this. They do not fit experimental timelines. They do not tolerate disturbance. They exist on scales, temporal and spatial, that challenge traditional scientific approaches. And yet, their existence expands the boundaries of what life can be. They show that size does not require multicellularity, that ecosystems can exist inside single organisms, that toxicity can be managed through architecture rather than avoidance, that stillness can be as evolutionarily powerful as movement. These are not curiosities at the edges of biology. They are alternative answers to fundamental questions about survival. As exploration technologies improve, more xenophyophore species are being discovered, each with variations in form, material, and habitat. This diversity hints at a long evolutionary history unfolding quietly, largely unnoticed, beneath kilometers of water. A lineage that has persisted not by changing rapidly, but by fitting perfectly into a narrow ecological niche. There is also a broader implication. When scientists search for life beyond Earth, they often imagine familiar pattern cells dividing, organisms moving, ecosystems cycling quickly. Xenophyophores suggest another possibility: life that is slow, spatial, chemically embedded, life that does not announce itself through activity, but through presence. They remind us that biology is not obligated to be obvious in the end. Xenophyophores do not offer closure or neat conclusions. They offer perspective. They show how much of life's potential remains unexplored, not because it is rare, but because it unfolds beyond the pace and scale of human attention. In the deepest parts of the ocean, life continues to experiment quietly. And xenophyophores stand as evidence that the rules we think define biology are, in many places, still being rewritten in the dark.